The identification of small molecule protein interactions is essential for the research and development of drugs, as well as for furthering our understanding of the pathological mechanisms underlying various diseases. This method facilitates the high-throughput biopanning of drug-recognizing peptides and global validation of drug binding sites on proteins for small molecule drugs of interest.
To prepare a QCM sensor chip, attach a ceramic sensor chip onto the oscillator of a 27 MHz QCM apparatus and record the intrinsic frequency in the air phase before small molecule immobilization. After the recording, detach the chip and carefully add a 20 microliter drop of a 1 millimolar small molecule derivative solution in 70% ethanol to create a self-assembled monolayer on the gold electrode of the sensor chip. Place the chip into a petri dish lined with moistened tissue protected from light for 1 hour at room temperature, before gently washing the electrode surface with ultra-pure water. Dry the chip with a gentle application of air and load the chip onto the QCM apparatus. After 1 hour, record the reduction in frequency in the air phase to measure the amount of the small molecule that has been immobilized.